Nucleotides are the building blocks of nucleic acids. Nucleotides are responsible for many key functions, including our energy currency. They can act as chemical links between different processes, and they are also the key components of DNA and RNA. And we know DNA to be the key information holder and transmitter within a living organism. So when we take a look at a nucleotide, we have three main components that are true for all nucleotides. So we have a nitrogenous base. Nitrogenous base simply refers to a compound that consists of nitrogen. Then we also have a pentose. A pentose is essentially a 5-carbon sugar. And then we have one or more phosphates. Now, if we take a look at this general nucleotide, we can see that we have a pentose. It's a 5-carbon sugar. We have our nitrogenous base, which is linked over here at this carbon. And we have our phosphate linked over here. Now, what's important to note in this depiction is that our phosphate is bound at our 5' carbon end. Now, if we remember from high school biology, we always remember that DNA runs 5' to 3'. So what does that mean? That means essentially that our phosphates are bound at the 5' end, and we see our nucleotides linking at the 3' end. Okay, so essentially the key distinction is phosphates bind at the 5' and we have the other nucleotides binding at the 3' end and so forth. Now, there's another term that we often confuse, and that is nucleoside. Now, the difference between nucleoside and nucleotide is that within a nucleoside, we do not have a phosphate. So when we only have our sugar and our base, that is a nucleoside. Once we attach our phosphate, it becomes a nucleotide. Now, taking it one step further, when we talk about nitrogenous bases, there are two main types of nitrogenous bases, and those are the pyrimidines and the purines. Now, these are the parent compounds. And we see various derivatives of these compounds within DNA and RNA. Now, some key points to make note of is within a pyrimidine, we have only a single ring, whereas in a purine, we have 2 cyclic structures. So it's important to also make note of the numbering over here. Here, this bottom nitrogen is number 1, and then we see number 2, 3, 4. But over here, this bottom nitrogen is a 3. And this is going to be very important when we talk about the linkages to the sugar. So if we take a look over here, we can see that in our sugar, at carbon number 1 is linked to our purine or pyrimidine, and it is going to be linked to either, if it's a pyrimidine, it's going to be linked to this nitrogen. If it's a purine, it's going to be linked over here. Okay, this is very important. Now, another thing to make note of is that when we are labeling our nitrogenous base, we are simply saying 1, 2, 3, 4, 5, 6. But in order to distinct it, to make it different from the pentose, when we label the pentose, we use prime. So we use 1', 2', 3', 4', just to make them a little different in our numbering system, to distinct them. And that's essentially what a nucleotide is, the difference between a nucleoside and our 2 parent nitrogenous bases. But now let's take a look at our derivatives of these 2 parent compounds. So here we have adenine and we have guanine. Adenine and guanine are our purines. And remember that our purines are structures that have 2 cyclic structures within them. So if we recall, we can remember that this nitrogen over here, it's nitrogen number 9, and this nitrogen over here, it's nitrogen number 1. And I can write that out. So this one was number 9, and this one was number 1. Now, the distinction between adenine and guanine is that over here, we have that amino group up here, but in guanine, we have a double bound oxygen instead, and we have our amino group at this carbon over here. Now, when we take a look at our pyrimidines, we see something different. We see actually 3 different compounds. We have cytosine, thymine, and uracil. Now, you may ask, why do we have 3 pyrimidines, but we only have 2 purines? Well, when we talk about DNA and RNA, so we have RNA. Within RNA, we have A, we have G, we have cytosine, but we do not have thymine, and instead we have uracil. And when we talk about DNA, in DNA we have adenine, we have guanine, we have cytosine, and we have thymine, but no uracil. So essentially, these 2 are the distinct characteristics of RNA and DNA. RNA has uracil but no thymine, and DNA has thymine but no uracil. Now, if we take a look at these pyrimidines, they look very similar to one another, but what's important to note is that they all have that double bound oxygen. The key structure, the cyclic structure, is the same. Cytosine has that amino group on top. Thymine instead has another double bound oxygen, so does uracil. So if we were to distinct thymine and uracil, we would say that thymine has that extra CH3, whereas uracil does not. Okay? So it's important to know the differences between the pyrimidines and the purines, since they all look very similar. But what's important to note, adenine and guanine, they have 2 cyclic structures, whereas the pyrimidines, they only have a single cyclic structure. Now here we can take a look at our DNA and RNA and how they differ. So we can see over here then in our deoxyribonucleotides, we have A, G, T, and C, but in our ribonucleotides, we have A, G, U, and C. Now what's really important here to make note of is that in our deoxyribonucleotides, so in our DNA, if we take a look at our pentose sugar, here at this carbon, we do not have an OH group down here. We actually have a hydrogen. But if we take a look at our RNA, we have an OH group at that carbon. And that's the key distinction between the pentose. Here we do not see an OH group, but here we see an OH group. And this is going to be very important later on when we discuss why RNA is more unstable compared to DNA. So that's the difference between the sugars, and the other difference was that we have uracil in RNA, and in DNA we have thymine. Now, the last thing I want to make note of is that we are linking the sugar and the base via a beta linkage. So remember that beta linkage always refers to pointing up, whereas alpha is pointing down at this position. So we can see the bond forming upwards, so it is beta, okay? If it was downwards, it would be alpha. So it's a beta linkage, and now we have to make note, is it at the 9th nitrogen or is it at the 1st nitrogen? Now remember, if it is a purine, purines have 2 rings, and this nitrogen is actually nitrogen number 9, so this is a beta linkage at the 9th nitrogen. But if we take a look at a pyrimidine where the 1st nitrogen is binding, this would be a beta linkage, but with nitrogen 1. And that is the difference between the linkages. We either have beta 9 or we have beta 1. And that is essentially an intro to nucleotides. We will go into more detail later about the specific bonding and characteristics in more detail in another video.